Hello, in this video we will discuss about the Young cycle. The Young cycle is basically used for the biosynthesis of ethene. So that is why it is the plant physiology we are understanding and the biochemistry. So ethylene is a gaseous, basically it is the gas and gaseous stage, and the ethylene is a gaseous colorless hormone, and it is important for ripening of fruit. So remember the importance about the ethene is the it is used for the ripening of fruit. So it is used for the ripening of fruit. So basically it is present in the plant, and the plant leaf will produce the ethene as well as other basically is the production from the uh, fruit. So here is the photosynthesis require the carbon dioxide and water. So water will split to produce oxygen, and this chemical reaction during the allied reaction is the photosynthesis to produce glucose. That glucose is used for the development, and on the other hand development and the ripening and the synthesis of the starch. So the ethylene is a synthesis in flowering plant from methionine via a cyclic pathway, which preserves the methyl thio group of methionine, which using ATP. So it is using ATP, and two notable intermediate in the cycle are S adenosyl methionine, and number 2 is the 1 amino acyl propane 1 carboxylic acid. So let's begin to understand with the explanation about the Yang cycle. And basically is the methionine. We will start from the methionine, but we can understand the cycle. So this is the methionine contain a CH3S, CH sulfur, which contains CH3 and CH2COOH, COO, and amine group. So this methionine will enter in the cycle, will convert through a adomate synthase enzyme. Adomate means the adenosine methionine. So adenosine methionine synthase enzyme use, and the ATP will use will produce the pyrophosphate, and the inorganic phosphate group also released. So it means the total phosphate group will not attached. In this way, the adenine formation here will addition, and in this way, the S-adenosyl methionine will be formed. And in this way, you can understand this. Basically, the addition of the adomate will lead to synthesis, because the adomate addition will lead to synthesis of the S-adenosyl methionine. And in this way, the aminoglycopropane carboxylic acid ACC synthase enzyme used to produce the aminoglycopropane carboxylic acid. This aminoglycopropane carboxylic acid will be formed, and the further reaction will produce the ethene or ethylene. So in this way, first of all, we should understand about the cycle. So in this way, here is the S-adenosyl methionine will also convert in the presence of ACC synthase enzyme to 5' methyl thioadenosine, because the adenine is attached with the carbon number 1, while the 5' side with the contain a methyl. So the methyl group with the you can understand. So anyhow, basically the methyl thio will attached, and in this way here is the 5-methyl thioribose formation after the releasing of the adenine. So in this way, the splitting and release the adenine will remain the 5' methyl thioribose. In this way, the ATP will use to release the pyrophosphate and inorganic phosphate. Again, that's process. And in this way, the 5 methioribose 1 phosphate will be formed. 5 methioribose 1 phosphate basically produced. And in this way, the oxygen will use to release the HCO2 as well as the phosphate group with containing 2 proton, and in this way it will be released to form a alpha keto and alpha keto gamma methyl thiobutyric acid. This contains a CH3S sulfur and the CH2 to CH2 to CO to COO negative. It is the alpha keto methyl thiobutyric acid. And in this way, here is the amine group containing, here you can see this will be release the 2 carbon, and in this way to produce again the methionine. So the methionine is the amino acid, which that is also important, and it is not a non-essential amino acid will produce in the plant. So here is the inhibit ethylene synthesis through AOA, the AVG, which that inhibit the ACC synthase to inhibit the ethylene formation. While this is, let's begin to understand the promote ethylene synthesis. It will be through a fruit ripening response, flower senescence, indole acid response, wounding, and droughts and stress, as well as the flooding. These all factors which that will target and trigger the ACC synthase enzyme to produce aminoglycopropane carboxylic acid, ACC. And this aminoglycopropane carboxylic acid, which that will produce the ethene or ethylene when the ripening response, and it can be inhibit basically ethylene synthesis through a carbon dioxide increase and anaerobiosis, and temperature will 35 degrees Celsius. An ACC oxidase enzyme used to remove the one half oxygen by the fixation of the carbon dioxide. So carboxylation will occur, and in this way the HCN will be released, and on the other hand the H2O also to produce the ethylene. While also the aminoglycopropane carboxylic acid by the malonyl CoA addition will be produced into the N-malonyl aminoglycopropane carboxylic acid. So anyhow, this is the 2 product can be synthesized, and in this way the ethylene will be produced, and this ethylene is very important. But the synthetic ethylene can be prepared in the lab by this reaction. So ethylene basically is by the dehydrogenation of the ethane to produce ethylene. As a synthetic will be used ethylene by the putting in the box, and the unripened fruit will after the cover to take time to ripening a fruit. So the Young cycle and biosynthesis of ethene we have discussed in this video. I hope you make sense about this cycle, and if you like this video, please make sure to subscribe, like, and share. And this is the basically the Young cycle. So you can give the suggestion for understanding more better. Next video we will discuss about the signaling pathway of the ethene. So but for this time, thanks for watching. Bye.